Welcome to the RevGenetics podcast. We're thrilled to have you join us today. If you want to dive deeper into longevity science, remember, you can find the RevGenetics podcast on Spotify and on YouTube.
Today, we're digging in something really exciting. It's a study that kind of bridges two very different worlds of biology.
Right, we're talking about the genetics of supercentenarians. These are people living past 110 and how that connects to a potential treatment for a really tough, rapid aging disease.
Exactly. Hutchinson-Gilford Progeria Syndrome, or HGPS, it's caused by this one specific mutation.
In the LMNA gene, right. And instead of the normal protein, it makes this faulty one.
That's it. It makes an abnormal toxic protein called Progerin. And this stuff, it just accumulates.
And causes havoc, essentially. It messes up the cell nucleus.
Totally. It disrupts the structure, the function, leads to massive instability, premature senescence, basically, accelerated cellular aging.
Which is why children with progeria age so rapidly. It's heartbreaking. And the primary cause of death is cardiovascular, right? Heart problems.
Almost always. Heart attacks, strokes, their blood vessels just can't cope.
Now, there is an FDA-approved treatment, lonafarnib, and folks are working on things like DNA-based editing.
Absolutely. Incredible work. But those are longer-term plays.
Yeah.
What's needed, like right now, are ways to protect the heart and blood vessels immediately to give these kids more healthy time.
Okay, so that brings us to the other side of this story, the supercentenarians.
Yeah, and this specific gene variant, they often carry the longevity-associated variant, or LAV, of the BPIFB4 gene.
What's special about their cells?
Well, they just seem incredibly resilient, especially against the kind of stress and nuclear damage that Progerin causes. It's like they have this built-in defense.
And here's a key observation from the study, right? In the progeria patient cells, this BPIFB4 protein is actually downregulated. Its levels are low.
Exactly. It's likely suppressed because of the Progerin toxicity.
So you've got low levels in the disease, high levels associated with extreme health and longevity. The hypothesis seems pretty clear.
Could giving this longevity variant, LAV-DPIFB4, actually protect against the damage in progeria?
Specifically the heart damage. That's what they tested.
Right. They went straight for the heart because that's the critical failure point in each GPS.
So what's the first sign of trouble in the heart for these kids? You mentioned cardiovascular complications.
Yeah, often the very first thing doctors pick up on an echocardiogram, an ultrasound of the heart, is something called diastolic dysfunction.
Okay, diastolic dysfunction. Break that down for us.
So your heart does two main things. Squeeze blood out, that's systolic function, and then relax to fill back up, that's diastolic function.
Breathe and fill. Got it.
With diastolic dysfunction, the heart muscle gets stiff, really rigid. It just can't relax properly between beats.
So it struggles to fill up with enough blood for the next beat.
Exactly. Like trying to fill a stiff old water balloon, you just can't get the volume in efficiently. And eventually, that leads to heart failure.
And they had a mouse model for this, one that develops this specific problem.
Yes, a specialized mouse model that mimics these early cardiovascular signs. And sure enough, if left untreated, these mice developed a pretty severe diastolic dysfunction around 7 or 8 months old.
How did they measure that stiffness?
Using those echocardiogram metrics, they saw a decreased EA ratio and a higher EA ratio. Basically, these numbers tell you how efficiently the heart is filling and how high the pressure is inside during that filling phase. Bad ratios mean a stiff, struggling heart.
But the squeezing part, the systolic function, was okay at this stage.
Yeah, in this early model, the squeeze was still strong. The problem was all about the relaxation and filling.
Okay, so the target was clear. Fix the stiffness, improve the filling. How did they deliver the LAV BPIFB4 gene?
They used a gene therapy approach, a single injection carrying the LAV BPIFB4 gene packaged inside a harmless virus.
A viral vector.
Right. Specifically an adeno-associated virus, subtype 9 AAV9. It's known to be really good at getting into heart muscle cells.
Wow. So taking a gene variant from someone over 110 and putting it into these mice with rapid aging, did it work?
That's the amazing part. They watched the mice for 2 months after the injection.
And the results, the aha moment.
It was pretty dramatic. The treatment significantly slowed down those age-related heart changes they saw in the untreated mice.
So it helped the heart relax.
Yes, crucially, it preserved the heart's ability to relax. Those key metrics, the EA and EE ratios, they improved significantly. They looked much more like those of a healthy heart.
That's huge, because that diastolic dysfunction, that stiffness, is often the first major step towards heart failure in human patients.
Exactly. It suggests that treatment was hitting the right target, potentially preventing that initial decline.
Okay, that's the function. What about the heart tissue itself? Did it look healthier under the microscope?
They looked at that too. Histology showed some interesting things. The gene therapy reduced perivascular fibrosis.
Perivascular, meaning around the blood vessel.
Right. Less scarring around the coronary blood vessels in the heart muscle. Scarring there can make vessels stiff and impede blood flow.
Makes sense. Did it affect other scarring?
Interestingly, not really. It didn't significantly change interstitial fibrosis, which is scarring within the main muscle tissue itself.
So it seems quite targeted towards the vessels and their immediate environment.
That's what it looks like, yeah. Protecting the plumbing, you could say.
And did the plumbing, the microvessels, actually improve?
They did. The treatment boosted the microvasculature. They saw more small coronary arterioles, those tiny vessels feeding the heart muscle, and better coverage by smooth muscle cells, which support the vessel walls.
So healthier vessels, more of them, better supported, better blood flow overall.
Seems like it. Better function, less scarring around vessels, and improved microcirculation.
Okay, we need to get into the how. What was happening inside the cells to cause these improvements?
This is where it gets really interesting at the molecular level.
Yeah.
The therapy tackling cellular senescence.
Ah, senescence. These are the old, damaged cells that hang around and cause trouble.
Right.
Exactly. They stop dividing, which might sound good, but they start spewing out all these inflammatory and damaging signals, the SASP, senescence-associated secretory phenotype. They basically poison the neighborhood.
Like toxic, grumpy old cells?
Pretty much. And the LAV-BPI-FB4 treatment significantly reduced the number of these senescent cells in the heart tissue. They measured it by looking for common senescence markers like P16 and P21.
So it was clearing out the bad actors at the cellular level.
It certainly seems to have reduced their numbers, yeah.
Now, to make sure this wasn't just some weird mouse-specific effect, they tested it on human cells, too.
They did. They used fibroblasts, connective tissue cells taken directly from human HGPS patients.
And what did those patient cells look like in the lab dish?
Pretty much like you'd expect.
Yeah.
Signs of accelerated aging, way more senescence markers like beta-galactosidase, and they were churning out signals that promote fibrosis, that scarring process.
Like which signals?
One key one they highlighted was EREG, iparegulin, as part of the EGFR pathway, heavily involved in telling cells to produce scar tissue.
Okay, so the patient cells are already aged and pushing for fibrosis. What happened when they introduced the LAV-BPI-FB4 gene variant into these human cells?
It was like turning back the clock, cellularly speaking. It reduced those senescence markers, and importantly, it dialed down those profibrotic signals, including EREG.
So the human cells became measurably younger and less prone to causing scarring.
At a molecular level, yes. That's what the data showed.
Okay, this leads to probably the most critical finding, the real core of the mechanism. We know progeria is caused by that toxic progerin protein. Did this LAV-BPI-FB4 treatment work by getting rid of the progerin?
You'd think so, right? But no, that's the surprising part.
Wait, really? The treatment worked, but the progerin was still there?
Exactly. They measured progerin protein levels and the mRNA that makes it.
Yeah.
The LAV-BPI-FB4 treatment didn't change them. The toxic protein was still accumulating just as much as before.
How did it help if the toxin is still present?
This is the key insight. It seems LAV-BPI-FB4 doesn't stop the toxin production. It works downstream. It reduces the toxicity caused by the progerin. It helps the cell cope with the damage, makes it more resilient despite the presence of the progerin.
Wow. Okay, that changes things. It's not about eliminating the cause directly, but about building cellular resilience against the cause. Like putting up a firewall against the damage.
That's a great analogy. It's mitigating the consequences, acting as a protective factor even when the underlying genetic problem persists.
We want to take a moment to thank our sponsor, RevGenetics. RevGenetics has been dedicated to providing high-quality longevity supplements and supporting healthy aging since 2007. If you are looking for ways to boost your health span, check out RevGenetics today.
So yeah, this study is really the first clear demonstration that a gene variant from a super healthy, super long-lived human can protect the heart in a model of this catastrophic aging disease.
Protecting against scarring, against senescence, preserving that critical diastolic function.
Right. It maintained heart function in the face of ongoing progerin accumulation.
The implications seem pretty significant. This offers hope for treatments that could work now, doesn't it?
Absolutely. While we wait for complex genetic cures like base editing to become routine, therapies like this boosting resilience could be a crucial bridge. They could extend health span, keep the heart functioning longer.
Giving patients more time, better quality time.
Precisely. And you have to think bigger picture, too. This is just one longevity gene variant, BPIFB4.
There are likely others hidden in the genomes of centenarians and supercentenarians.
For sure. The genetics of these exceptionally long-lived people. It's like a natural library of protective mechanisms. There could be secrets there for treating all sorts of age-related decline, especially in the heart, far beyond just progeria.
It really shifts the focus towards learning from natural resilience. This has been an incredible deep dive connecting extreme longevity with the potential therapy for rapid aging. Thanks for unpacking all that.
My pleasure. It's truly fascinating stuff. And maybe a final thought for you, for everyone listening, to chew on. The evidence suggests this BPIFB4 variant works by bolstering some really fundamental cell maintenance jobs. Things like keeping the nucleolus healthy, making ribosomes correctly, preserving telomeres. All things Progerin screws up badly.
Right, the basic housekeeping of the cell.
Exactly. So if just this one longevity pathway offers such powerful protection against a disease driven by disrupting those basics, what other fundamental protective systems, maybe ones we haven't even found yet in centenarians, are we potentially missing? What other natural defenses could be boosted, not just for rare diseases, but for common age-related decline? What resilience are we lacking?
Something to definitely think about. How do we find and boost our own built-in resilience? Fantastic stuff. We hope you enjoyed this dive into the connection between supercentenarian biology and progeria research. Thank you for listening to the RevGenetics podcast.
And finally, a big thank you again to our sponsor, RevGenetics, who has been committed to advancing longevity through supplements since 2007. Until next time.